So I want to talk about DNA and how random it is and how to analyze it. So we talked last time about human DNA is actually 3 billion base pairs. So that's 3 times 10 to the 9, and every single cell has the same amount of DNA and has the same type of DNA. And we talked about how it's got to be supercoiled in order to get it in there, and it's got to be nailed down with histones in repeating units, which we call nucleosomes. So we have 3 billion base pairs in every single cell. Wow, that's a lot. But the really interesting thing about it is most of it is really just junk DNA. So really about only 10% of the DNA is considered to be usable, or what we consider to be genes. And genes are things which produce proteins. So the other 90% is really just junk. As far as we know, it serves no purpose. There's been a number of ideas of what it could be doing. But for now, we're only concerned with the 10%, which are genes. How many genes? Well, probably about 20,000 genes. Now, the estimate of number of genes has really come down. I remember at one point it was like 150,000 genes, they thought. And now we're down to 20,000. But we'll stick with that number. Now, here's the really interesting thing. You may think that we're all very, very different. And we are in some ways. We're all unique. However, the part that makes us unique is actually a very, very small part of the DNA. So if you take any two people and you certainly recognize them as being different, really, if you look at the percentage of DNA, it's a very small percent of 1%. So really, we're all like 99.9% the same as far as our DNA. Why would that be true? Well, I mean, you recognize right away differences, but they're really very trivial differences, the color of your eyes or the color of your skin or things like that. If you think about the big stuff that we have in common, the fact that we have a heart, the fact that we have 2 lungs, the fact that we metabolize glucose, and these sort of things, they're all the same, really. So most of our DNA is the same. In fact, we actually share 98% of our DNA with chimps. So you definitely recognize a chimp as being different than a person, but 98% of our DNA is the same. 92% of it is really the same as a mouse. So almost all our DNA within the same species is just about the same. It's only a very small percentage which is different. How do we know this? Well, the major technique that's used to analyze DNA is what we call gel electrophoresis. Okay? Now, gel electrophoresis is called that because you pour a gel, just like gelatin, jello, and you pour it in a frame, and then you load up DNA samples in the top here. And then you go ahead and you run an electrical current through there, right? So you go ahead and you run an electrical current like so, and this causes the DNA to actually start moving. And it actually separates based on size, right? So the big ones start here, and then the smaller ones go down here, and you get these really interesting patterns of bands, little pieces of DNA that separate out there. And you can see that sometimes the sample will have bands in common, and sometimes, oops, that one there, it's missing, it's not over here. And that's how we can compare whether a DNA piece is the same or different. Now, we use enzymes, which we call restriction enzymes, and those enzymes cut up the DNA, and it creates all these fragments which then get separated. But wait a second, let's go back to what we said before. Gel electrophoresis really isn't a great idea to tell us the difference between one person's DNA and another. So you always hear about these DNA tests on CSI and so forth, but if our DNA is 99.9% the same, most of the patterns here are going to be exactly the same. In fact, it would almost be impossible for you to find where is that one little band, right, which is different from everybody else. Because remember, you're going to have 3 times 9, you're going to have 3 billion bases of DNA, so you're going to have hundreds of millions of bands here. So how do you tell the difference? Here's the really brilliant idea that somebody came up with. Since most humans have all DNA in common, the patterns are pretty much identical. How do you tell the difference? You don't look at what we have which is different, you look at how much we have in common. Because we know we all have big hunks of DNA in common. Now here's the interesting thing. There's several different types of DNA that we have in common. They're called variable number of tandem repeats, VNTR, short tandem repeats, STR, satellite DNA. What is satellite DNA? That's the type that we find in the centromeres, right? And then we have mini satellite DNA. Let's just talk for a moment about this one over here, because this one I think is the one which will really answer the question. VNTR, variable number of tandem repeats. We all have the same sequences of DNA, but for some reason in our junk DNA, 90% of our DNA, these big hunks of DNA just keep repeating. So you have a certain base code, and it just keeps repeating over and over and over again. But some people might have 1, 2, 3, 4, 5, 6 of them. Some people might have 4 of them. Some people might have 3 of them. Some people might have 5 of them in a row. So what you do on gel electrophoresis is you look at these areas we have in common, these VNTRs, and you run them on a gel and you see how many we have in common, and that tells us if we're different or if we're the same people.